Howdy, my name's Seth Murray. I'm a professor in the Eugene Butler Endowed Chair at Texas A&M University. I'm a corn breeder, and I've been a corn breeder here at Texas A&M for about 12 years. My background's in molecular quantitative genetics, and I do run an applied corn breeding program. So I try and apply the theoretical knowledge and skills I had from graduate school and my training into my breeding program. So what I've been asked to present to you today is on some molecular quantitative genetic types of activities that would further a molecular plant breeding program. And I don't know what everybody's background is going to be who ends up watching this video, but most of what I'm going to present today, I actually cover in my PhD level class for senior graduate students here in our department. So I'll skip over some of the technical details and try and maintain a high level overview, but this really is a semester long set of topics to get through. So I'm going to begin giving you this overview. Then I'm going to talk about QTL mapping, or quantitative trait locus mapping. I'll talk a little bit about genome-wide association studies and mapping in those. We'll talk briefly about marker-assisted selection in a breeding program. And then we'll move to genomic selection and phenomic selection. And what I'm showing you in the background here is actually one of our cornfields with a drone or a unoccupied aerial vehicle, or UAS, flying over it. And my breeding program is rapidly moving completely away from molecular breeding tools into phenomic breeding tools for a number of reasons. And so a lot of the same techniques and background that you need for genetic mapping, you can use in phenotypic mapping from UAVs or drones. But we'll cover that as we get a little bit further. So why molecular breeding? I assume anybody who's going to pay attention to this course probably already thinks that they know why. But the main reason that molecular breeding began was because plant breeding is slow. It's labor intensive. It's a lot of work, and here in Texas, it's hot and miserable to be out taking notes in the field. So is there any way we can speed that up through technology? And for a long time, people believed that genetic markers could help us do that. So for those of you who aren't plant breeders, I'll just point out that in 7 years, we went from something like the BlackBerry to the Samsung S9 Note. And as a Samsung fanboy, I'm going to put that up there. But if you're an iPhone user, I'm sure you can find a similar dramatic transition. In that same period, most optimistically, we might go to one new variety. Okay, so it takes 7 years in terms of the breeding process to develop a new variety of corn. So we're looking for tools to expand that. Now, how do we simplify this and make this theoretically a faster process, a better process? Well, we can think about the breeder's equation. And the breeder's equation is basically how do we get genetic gain over time? So for those of you who are really focused on molecular markers and just using molecular markers for molecular marker's sake, that is not what this lecture is going to be about. It's going to be about how we can actually speed up plant breeding by using tools, including molecular markers. So the genetic gain over time is our goal. We want to make the maximum amount of progress in the shortest amount of time. And that's going to have to do with a number of different factors here, like the selection intensity. And how do we overcome that? Well, we grow more plants, and we select the same number of plants by growing more plants, and then we have a greater selection intensity. Selection accuracy, we need to measure our plants better. Now, for a long time, people believed that genetic tools would help measure plants better. It would help break down some of that variation and allow us to have discrete units we can select on. And we'll cover whether or not that's really been all that true or useful. Additive genetic variants. So how much genetic diversity is there? So for those of you who are familiar with plant breeding, if you had a whole field of a single variety, a single inbred line, and you try and select the best individual from that, you're not going to make any progress because there is no genetic diversity. Whereas if you go into a wild population, there may be lots of genetic variants there, but mostly what you're going to select out is trash, stuff that'll lodge or shatter, so it won't be useful anyhow. And then, of course, the years per breeding cycle. So one of the ways we've been able to shorten the breeding cycle is by using off-season winter nurseries. Using either a glass house or using something in a different hemisphere or in a different part of the world. We also can improve our years per breeding cycle by selecting on things before we even plant the plants. And so that's where molecular markers and molecular breeding are very useful. So just to give you a little picture of where we're going here, this was part of technically a QTL mapping study, but we used genome-wide association approaches to map some simple traits in a four-way parent magic population, which we will cover briefly. And so what we can see here is on the y-axis is the significance. On the x-axis is the genetic markers across the chromosome in maize. And we were able to, by combining our phenotypic data, that is measurements for whether or not the plants were red cob or white cob, or whether the grain was white or yellow, or blue or white or yellow, we were able to identify regions of the genome where the variation for these traits is conditioned. So that's the whole point of QTL or GWAS linkage mapping. QTL linkage mapping or GWAS is to associate genetic markers with a phenotype to identify the location of where genetic diversity controlling that trait resides. Again, if we were to try and do this on a population of inbred lines, it wouldn't work. The reason being is there's no genetic variation in inbred lines. There should be no segregation in phenotypes, nor any segregation in genetic markers. And so it would be a failed experiment. And again, if we take the other extreme and select wild individuals, there's going to be so much other variation going on that it'll be difficult to hone in on what we're most interested in here. So I want to cover a couple generalized methodological waves and generalizations in molecular plant breeding. So it was about 1986 when QTL linkage mapping started. There were tomato and maize populations, and a lot of the people who did this initial work are in the National Academies of Sciences now, or quite world-renowned. Now the goals of QTL linkage mapping are as follows. To find loci. Now note that it doesn't say finding genes, because QTL linkage mapping does not allow us to resolve in almost any population to the level of genes. We find regions of the genome, which we call loci, and then we can do follow-up studies such as map-based cloning to identify the genes that cause those sources of genetic variation. Typically, we're going to be using biparental families. That is, families of individuals that have two parents, which would bottleneck and restrict down the genetic diversity. We're going to be able to make inferences to specific populations. Okay, so one of the challenges, one of the ideas that people have that is generally proven to be false, is that if they identify a QTL in one population, then they can go apply it to different populations. And in some crops, that's more true than others. But in maize, it's very rarely true that if you identify a QTL in one population, it will actually work in another population or be causal. Now, the end goals of QTL linkage mapping have primarily, this is over the 40 or so years this has been occurring, have been to generate knowledge about the genome, about crops, about genetic inheritance, to generate publications, and you'll find lots and lots of publications on this topic. But the actual application of crop improvement has been pretty minimal. I don't think we can point to too many quantitative improvements in crops that are due to QTL linkage mapping. There have been a number of qualitative traits, a number of major genes that are often used, but not so much the minor effect quantitative traits. Now in 2005, we started seeing an approach of association mapping, or genome-wide association studies, GWAS. The goal in this was most definitely to get closer to finding genes. And these populations are typically very diverse, or more diverse than a biparental family. So we're taking advantage of the historical LD, or linkage disequilibrium, to help us get closer to resolving genes than loci. Now, one of the huge advantages of GWAS from my breeding perspective is that the alleles that you discover actually work across population structure. So that is, if you discover them in one population, they're much more likely to work in another population than in QTL linkage mapping. And then I think what we've often found, though, is that in the end, a lot of knowledge is developed, a lot of publications are published, but the actual application of crop improvement maybe hasn't quite been as great as we hope. So that puts us into the modern era, 2009, where we start seeing, to present, where we start seeing genomic selection or genome-wide selection, depending on who you talk to, come over as the main methodological approach. And this is the approach that industry is actively using, in the United States at least, and in Europe, and that really seems to be working. So in this case, the goal is not to find genes or find markers, it's to select the best individual. So it's much better aligned with an actual plant breeding objective. The populations, you can choose a lot of different types of populations. You really could use a biparental population, that's probably more common, but you could also use a diverse population if you wish to. Now, one of the huge helps for plant breeding is if you train your genomic selection model over material that's relevant to a breeding program, it should then be predictive to other material in the breeding program. Now, these estimates have to be updated every few years, and there's lots and lots of research that's gone into the best way to apply this. And I want to stay at a high level here. But again, the goal is to improve crops through this. It has generated a lot of knowledge, but I would say largely the people who have published on the methodologies and on the first few genome-wide selection studies had good publications. At this point, however, it's not very interesting to a reader because those genomic selection studies just tell you what the best individual is. There isn't broad knowledge that can be transferred from my breeding program to your breeding program in this case. And that's a major drawback for those of you who are in an academic or a job that publication is a primary goal. So really this has been a change from discovery or estimating effects to predicting the best genotypes. And that's really what we want to do in plant breeding anyway. We want to predict the best genotypes. We've also gone from simple trait measurements, and so a lot of the QTL linkage mapping material was often on fruit color or plant height. And as we're getting into prediction, we're considering value indices now, like, is there a value to sustainability that we can select on? Is there a value to yield or the seed size? Taste, nutrition, cultural preference. Okay, so why do we conduct QTL mapping? So what I'm going to do for the next, hopefully not more than 20 minutes, is focus just on QTL mapping, how we do it, what it means. Then I'll go a little bit into GWAS, and then a little bit into marker-assisted selection, and then to phenomic selection. So the goal here in QTL mapping is to identify the genetic control of the trait. So what's the inheritance? Is it inherited in an additive manner or a dominant manner? Where in the genome is it? What's the position? And what is it co-localized with? Which I'll show you an example of a little bit later. We also hope to identify molecular markers for marker-assisted selection. So you can't do marker-assisted selection unless you've already identified some loci to select on. In some cases, a much more academic topic is to identify homology with other genes or species in some type of comparative mapping approach, to get hints on genome organization, so these are not breeding goals, these two here, but then a breeding goal might be to select elite individual by predicting breeding value. That's a little less efficient through a QTL mapping approach than through a genomic selection approach. And then finally, there are a bunch of people who really focus on cloning QTL. So once you clone a QTL, there's a number of different things you can do, but this ends up being $1 million, literally $1 million US experiment to clone a QTL. And so our goal here is to take phenotypic variation that you see segregating in a population, identify the locus, identify the gene and the functional polymorphism that causes it if we're cloning a QTL. We won't talk at all about reverse genetics, but that was an ambitious idea that was much more popular, I think, 10 or 15 years ago, and largely didn't pan out. Okay, so what is a QTL? What does it look like? How do we get it? So in this case, I'm actually showing you simulated data. So in my graduate class, we simulate data in R, and then we test it to see how good you would actually be able to detect something in a real world situation. Because remember, if you run your experiment and you find a QTL, that does not mean it's real. It means there is a statistically significant detection of a locus there. But in terms of, is there something actually real there, or is it an anomaly of statistics? Is it an anomaly of your environment? Is it an anomaly of your markers? That's an open question. So if we simulate the data, we know what the truth is. In this case, I think I simulated an effect size for the big A marker of 5 centimeters. So the base height of these individuals, although they're randomly generated from a normal distribution, the base height is around 95 centimeters. It should be 100. And we can see the genetic variance of individuals with little a. Then individuals with the heterozygous genotype, big A, little a, show that the average height of those individuals is 105 centimeters. And then those with dominant big A, their height on average is 110 centimeters. But you can see that there are individuals in big A that are actually shorter than the tallest individuals in little a. And that's what we expect. We don't expect one gene to affect all of plant height. It's a quantitative trait. So that means there's many genes affecting it. And so what we're seeing here is the effect of, this is similar to seeing the effect of many cumulative quantitative trait loci combined together to create these phenotypes. Now, when we run this in a mapping software, and I apologize, this is a little bit old, is QTL Cartographer from North Carolina State University. This is one of the output files. And what this is showing in composite interval mapping, which allows you to connect different markers across the genome, is where we see the most significant effect, the most likely position for a locus affecting our trait. And so what we can see is there's a marker here at this position, and this is actually real data here from my PhD in sorghum. But we can see the effect of this marker here, and then it goes down a little bit for this marker, and then up a little bit for this marker, and that's just noise. None of that's real. This is our significance threshold, 2.5 log. And so anything above this we would say is a significant effect size. So then we see this effect drop off, and then it looks like there might be another QTL here, although it's just barely above the significance level. So this is an example of a QTL. We're combining genetic markers on the x-axis here and on the x-axis here with phenotypes, height in this case, and then it's been converted to a significance or a LOD score, likelihood of odds ratio score in this figure. Now, I can't emphasize this enough, and this is one of the biggest problems that I think people run into when they start looking at using molecular tools in a breeding program. And that is, most QTL are false. And at the end of this lecture, I'll talk about some of those reasons, but most QTL are not real. They are statistical anomalies that will never be repeated. And so there's ways to minimize this, but in this case of this, this is a simulation study by the famous Rex Bernardo that he did back in 2004. And we can see that this horrible ratio here where there's only one true positive and 57 true negatives. So that means there is no QTL. He didn't find a QTL. Here there is a QTL and he found it, but then there's 3 false positives. There's no QTL, but yet you find one, and 3 false negatives. There is a QTL, but you don't find one. And so this is actually quite common. And this is one of the problems with using real data to talk about QTL mapping. which is oftentimes we can find false positives or false negatives and not what we're actually looking for. So that's one of the reasons that simulation studies are useful for understanding QTL mapping. Now we need to talk a little bit about sample size and power, because the thing that I spend so long with my graduate students on is to not just use a recipe for QTL mapping or GWAS or whatever. It is a tool. Unlike all tools, it should be applied to the appropriate problem and appropriately. And so the first question is, what is your experimental goal? If your experimental goal is to clone a gene, then you should think about that up front and the amount that it'll cost. Now, I know a lot of people think they'll get lucky, but the data has shown most people will not get lucky and will not clone a gene that causes their trait. Now, if you're working in the disease field, you're much more likely to be successful. But in my experience, most people are working on trying to improve yield or improve something for farmers, drought stress, and to identify a gene for that is very, very difficult. So we need to be realistic about what our experimental goal is. Next, what is your heritability of a trait? And I don't have time to go into heritability in this lecture, but what is the heritability of a trait? So what does that mean? Well, if your heritability is 0, that means the variation for your trait is caused completely by measurement error or by genetic by environmental interaction or by other types of error. And if your heritability is 0, then there's no reason to go about QTL mapping or GWAS or anything really. If your heritability of your trait is high, you're much more likely to find useful variation from a molecular standpoint. How many QTL are involved? So the more QTL you have, the more individuals and markers you will need. So if you just have a single gene, if it's a single dominant or a single major gene, like we see in disease resistance, then you can get away using many fewer individuals, many fewer environments, and the markers will help you resolve the gene much more quickly. How large of the effect do you want to be able to find? Now, this is a really important question, and it goes back to your experimental goal. So in a famous study I'll talk about a little bit later, the nested association mapping study in maize, I think the largest effect was a day in flowering time. But most of the loci they found corresponded to 0.1 days or 0.5 days in flowering time. So if you have an effect, if your QTL causes a 0.5 day change in flowering time, how are you actually going to take advantage of that or measure it? You can't measure a 0.5 day flowering time difference. So it ends up being a statistical issue where you know it's there and you know it's real, but it doesn't have any practical value. So how large of a QTL effect do you want to be able to find? Or are you going to design your study so you can detect many small effect QTL and somehow pyramid them? What are the effects of your trait? So if it's dominant or if it's additive or over-dominant, all those things will change the sample size that you need. Is there any reason to believe that there's epistasis? Now, epistasis is the interaction between different genes. So if I have the big A allele on one genetic background, then we would not be able to detect it. And if I had the big A on another genetic background, we would. So if you have epistasis, just one epistatic gene can double the amount of individuals you need to look at to find your trait of interest. And we know there's epistasis throughout the genome. And that's one of the largest reasons that molecular quantitative genetics has not really worked out the way that people thought it would, is there's pervasive epistasis. So is there any reason to use a smaller than optimal sample size? So yes, with a caveat. If your goal is to write a publication or to graduate, then maybe that makes sense. If your goal is to conduct meaningful improvement in a crop, then no. I would say you'd be much better off taking the same amount of money and doing something more useful with it. So a couple reminders, and this is true for all molecular quantitative genetic work, is there is proper statistical preparation that you need to do. You need to find outliers. Outliers will have a huge effect. and will lead to many false positive and false negative QTL. You need to look for the normality of residuals. Okay? So it doesn't matter that your data is normal going into your model. What matters is it's normal coming out. What do I mean by that? Well, if you have one large effect gene, that large effect gene can create a bimodal distribution. And so what does a bimodal distribution look like? I'm going to attempt to use the tools in Zoom here. Bimodal distribution, if we have our graph and I showed you plant height, so we'll just call it H here, you would see something that looks like, well, it would make more sense to draw it like this. All right. So, so what we would see in a bimodal distribution is we'd see, so height should be on this axis, I'm sorry. We'd see some tall individuals and we'd see some short individuals. And there could be one major effect locus, let's say big A here, causing that difference between this average and this average. If that's the case, it doesn't matter that your data is not normal going in. What matters is the normality of your residuals. However, for quantitative traits, where many genes are involved, we typically will see a normal distribution that looks like that. Genotyping errors. Genotyping errors are extremely problematic for a number of reasons, but they will lead to many problems throughout analysis. You need a genetic map for linkage mapping, for QTL mapping. So you need to double check the genetic map is right. And genetic maps are an art. They are not a science, unfortunately. There is a reason that people have high expertise just at creating genetic maps. Now anybody can create a genetic map by putting it into some software, but understanding what makes a good genetic map or a bad genetic map is an art, and it's far beyond the scope of this presentation. And then check for equal variance. Okay. What do we need to map a QTL? So we need a controlled segregating population. So biparental populations are most common. So using an F2 or recombinant embryo lines, using a backcross population. However, there are some other populations that work with other techniques. So when we start talking about association mapping or GWAS, we're talking about diverse populations. So for this component of QTL mapping, we're going to talk about biparental populations. We need heritable variation, but there's a note here. It doesn't matter that the parents are segregating in their phenotype. What matters is the progeny are. So transgressive segregation is where the progeny have effect sizes more extreme than either their parents. So you cross a medium plant with a medium height plant, and you get plants that are shorter and taller than both the parents. That still is heritable variation that can be mapped. So you're going to need phenotypic data and good quality phenotypic data. So that means measuring the plants in the field, typically. You're going to need a molecular marker-based linkage map. And then most importantly, you're going to need some recombination. And that recombination is going to disassociate those molecular markers and the phenotype so that you can find your locus of interest. Okay, so a simple mapping strategy. We're going to look for, and I covered this before, actually, I think it probably is more useful if I go back to this figure here. We're going to apply our genetic markers with our phenotype, connect those 2 points of information, and then we can just run a simple regression here for single marker analysis. Now, as you start getting into hundreds or thousands of genetic markers, this is not an efficient strategy, and interval mapping or composite interval mapping are more appropriate. The other thing I forgot to point out here is that we have a sort of a fit to the line, so we have an R squared. We also have an effect size, and so our effect size in this case is 5. So each allele each large A allele increases the height on average by 5 centimeters. Okay. The other thing I'm not showing here is the significance. So is this a significant test? That's how we find whether or not there's a QTL. Okay.
I'm not going to go over the actual details on how to run this analysis, because that's part of what the students are going to do. But just the basic, most simple QTL analysis. We're going to code our genotypic data. Computers aren't so good at reading big A and little a, so we code our parents as zeros and ones. And then missing data would be treated as 0.5, because we don't want to bias it one way or another. We don't need a genetic map for the single marker analysis. So we need to prepare our phenotypic data by getting a best linear unbiased predictors, or some summary. We don't typically run each replicate separately, and averaging replicates within or especially across environments is definitely discouraged. Running a basic statistical model to get best linear unbiased predictors, or BLUPs, is a better, or BLUs, best linear unbiased estimators, is a better strategy. And then we're going to simply regress our genotypes onto the phenotypes. So we could use a t-test or an ANOVA. So it's important to remember that the marker is the repeated unit or predictor, not the genotype, right? And then significant genetic marker means the marker is linked to a quantitative trait locus, right? So if we found that this was significant, then we could say that a QTL was detected. And then the regression slope is the effect size, right? And in this case, I simulated a difference of 5 centimeters from each. For those of you who are familiar with R, I used the R norm function to generate normal random values. And what we can see is little a had a mean of 100. The heterozygote had a mean of 105. Big A had a mean of 110. And then there was variance and a number of observations associated. So here's your 1 to 2 to 1 ratio that you would expect from a single locus. Now, significance and effect are two separate things. So we can have an effect size at every single marker, even if it's not significant and even if it's not a QTL. The effect size is what the model predicts the effect of each big A is. Significance is going to depend on how big that difference is and how many observations you have, right? So if we want to be able to say there's a QTL and we need significance, then we probably need a lot more observations than 2 or 3, right? Okay, so I'm going to go over some examples, and again, this is all simulated data, but just to show you what the difference between an effect size and significance would be. And you can see up here, simulate random normal variables, 50 individuals of the recessive, 50 individuals of the dominant, 100 heterozygous, the height's the same, variance of 3, right? And so in this case, we almost perfectly estimate the effect size at 10, right? Going from 100 to 110, we estimate 10. The significance is very, very high in 200 individuals with an effect this big. Now, when our variance is larger, when our overall variance is larger, in this case 17, we can see that the effect size, if you look at the y-axis here, this goes from 95 to 115. Here it goes from 60 to 140. And so the overall effect size is not going to be estimated as well because there's a lot of other sources of variation here. That also means our significance will be far less. In fact, we might not even declare this as a QTL if we were using 0.01 as a cutoff. Now, if we have some type of dominance effect, that's also going to change our effect estimate a little bit and our significance, but in general, the additive model still does pretty well. And if we drop our number of observations to 1/10 of what it was, even though we have a very similar effect size, right? We're simulating 5, 10, and 5 individuals. Our effect is not estimated as well, and our significance is much, much lower. And I'm not going to go through all of these, but you could play around with this a little bit and use this to predict what size QTL you are hoping to capture in your analysis. I'm going to take a pause for water right now and turn on the lights. Okay, what I wanted to show you here, and this slide was borrowed from Ed Buckler, and this was part of Schoen's publication in 2002, is the importance of how marker density and the actual QTL effect can change your estimate of the QTL. So if you imagine a genome has millions of or billions of base pairs, the causal nucleotide that causes the phenotype you care about, you are very unlikely, in fact, it's almost impossible that you would measure it. So instead, what we do is we sprinkle random markers throughout the genome and use those to track segments of the genome. So what this little figure here is showing is that you have some marker, so big A or big M and little m, and a QTL, big Q is our QTL, little q is the null effect. And there's some probability of recombination here. And as the distance between your marker and your QTL grows, you actually have lower effect, you estimate lower effect sizes and have less ability to detect the QTL. That is because recombination is breaking those apart. So in fact, if the marker and the QTL were on opposite ends of the chromosome, even though that chromosome has the marker, because there is generally complete recombination between the two ends, you would not find that QTL. Okay, so I won't go through this calculation, but it's important to remember that you're almost never actually going to have a marker that is on the QTL. There's always going to be some form of recombination, and that's going to change the effect size that we're actually able to estimate. Okay, I think in the interest of time here, I'm not going to be able to get in the weeds too much, but I want to show you a little bit about sample size again. And in this case, it would be unbalanced data, right? So in this case, I'm showing you a height of the 3 genotypic classes. And if we had the same number of individuals, let's say you have the ability to grow 200 individuals in your program and screen with a genetic marker. So if you used an F2 population in this case, that is 50 dominant, 50 recessive, and 100 heterozygotes, your significance would be at this level, 1.6 times e to the negative 5. If you were able to use recombinant inbred line individuals, where you have a much more distinct difference between dominant and recessive variants, so 100 big A's, 100 little a's, your significance would be much better. So the note on increasing power here is that recombinant inbred lines are always going to be more powerful than F2 individuals, just because that heterozygous class really doesn't add a lot. So there's a number of different populations you could use. F2s, recombinant inbred lines, doubled haploids have some advantages, back crosses have some advantages. Advanced intercross lines like the intermated maize population have some advantages. Or dihybrid rills. So sometimes you don't have the opportunity to develop inbred lines. And so we sometimes see F1 crosses or family types of analyses. Now, I don't particularly do a lot of these myself, so I can't go into too much detail here. But if you had, I think cassava requires outcrossing. I don't think you can have inbred cassava genotypes. Then you might use a method like this. And of course, a lot of trees also use this because they just don't have the time to do the inbreeding needed. Okay. So let's go back to goals here. So I want to go between high-level overview and then some specifics, but not too specific, so that we can sort of understand what we're trying to do. So when we think about the type of populations we're going to use, there are 2 common goals. So the first goal, which I think most people claim they want, is to find a QTL that will improve their trait of interest for breeding. And so if that's your goal, then you actually need to do the study on an elite by elite cross. That is the best line you can find by the best line you can find. Now, what most people actually do in terms of what the published literature shows is trying to find the cause of trait variation. And to do that, you want to maximize this difference. And so you would choose an extreme low parent by an extreme high parent. Now, oftentimes, these types of crosses are not done in breeding material. And because of that, the QTL that you find in these type of crosses do not apply in a breeding population. Why? Because the breeding population has already been fixed for the better allele in this case. So it's really important that before you even start this experiment of QTL mapping, you think about the population you want to use and align that to your end goal. And I don't think people do that quite enough. So what does a genetic map look like? This is an older publication, not one of mine, on tropical japonica rice cultivars. And what they're showing here is 12 chromosomes, genetic markers that have been linked together. And if you remember, I said this is more of an art than a science, linking these markers together to create a realistic and meaningful genetic map. And then you can see QTL are assigned to various positions. And these QTL are not assigned to a single marker. They usually have an effect size that covers multiple markers. And that just reinforces why we call these quantitative trait loci and not genes. We're nowhere near resolving to a gene level. And in this case, there might be 1,000 genes under these QTL, any of which might cause that effect. It might not even be genes. It might be some other non-coding piece of DNA. So I'm just going to briefly go over interval mapping. So what I showed you before was single marker analysis. It's the simplest, most straightforward type of genetic mapping. But interval mapping, I'm going to skip over all the equations, and because there's software that basically does this for you. You input your genotype, and you input your phenotype, and you are able to calculate where in the genome the QTL is. And what an interval mapping situation does is it uses a much more complicated algorithm to say, okay, we know that B and C genetic markers are 35 centimorgans apart. And so given some effect size, we can test for sort of pseudo genetic markers that may occur here, right? Based on where other recombinations are happening, we know which individuals are more likely to have a recombination here, a recombination here, or no recombination in this interval. And so that's what the goal is in interval mapping, is to test QTL at all markers and between all markers. And that's where I'm not going to go through the equations, but that's where you start to see those nice smooth lines. A lot of people ask what a LOD score is, and to really understand the LOD score, we'd have to go through all the math. But in general, what we're doing with a LOD score is we're taking the negative log of the likelihood of the estimate under the null hypothesis, right? And that null hypothesis is saying that there is no QTL, right? And the variance that is occurring there, and taking that over the likelihood of the mean and the effect size, right? And the variance if there was a QTL. And so our hypothesis that there is a QTL is based on some likelihood. Now, trying to simplify this, I think what it really should reinforce for you is it does not tell us that there is a QTL. It tells us what the likelihood is that there's a QTL. And we set some boundary to say whether or not we believe that's significant or not. Okay. So we won't go over the permutation test. I mean, we can get in the weeds here, but I really would like to get through to some of the other more high-level things that would be more useful to you. So this is an example of interval mapping. The genetic markers, again, are on the x-axis. The LOD score, the likelihood of odds ratio, is on the y-axis. And we set our significance threshold based on a permutation test to correspond to 0.05 or something. So we will then say any peak that crosses over our significance threshold is where we believe there's a QTL. Now, there's a common way to report the interval that's included here, because note that this marker here, TXP120 and TXA276, both would be significant. And that's a long span of the genome. So we see the peak here and we say that is the most likely spot for the QTL to be, but there is no guarantee it's there. And oftentimes it will be off to the side. So we can talk about the 1 LOD support interval, that is 1 LOD less than the peak, or the 2 LOD support interval, which is 2 LOD less than the peak. Now the reason this is important is because if you're gonna use some type of marker to track your locus going forward, we really would like to use markers that flank the QTL. And if we use the 1 LOD support interval, it's more likely that the QTL actually resides outside of that interval. If we use the 2 LOD support interval, it's much more likely that there'll be a recombination event between there and our marker and our QTL will dissociate. And so this is just another important consideration and understanding to have in using these tools in marker-assisted breeding. So the interval mapping, the main advantage is it's using the genetic map, which combines additional types of information to give us more power. But there's a lot of problems with this, and I don't want to go into the specifics here. There is a famous study by Bill Bevis called the Bevis effect. He came up with something that is now referred to as the Bevis effect, again with simulation studies. And what he showed is that the QTL with a low power detection have effects that are often really overestimated, especially in F2 populations and in small populations. Now, why is that important? Because most of the loci we're going to find throughout the genome, I'm only showing a large effect locus here, but oftentimes you'll see some other smaller loci bubbling up. Many of those loci are likely to be false QTL. Okay, so these ones with the largest peak and somewhere near the peak is our most likely position for a true QTL, but that doesn't mean it is one. I'm going to skip over this, and we can talk real briefly about nested association mapping, because this bridges QTL mapping and GWAS really well. So the nested association mapping population in maize was first developed and published on back in about 2008. And what they did in this case was cross 25 different diverse lines to B73, which is the most famous US maize inbred line. And then they developed 25 different recombinant inbred line populations and used these in combination. So what this is showing is that the F1, you have one chromosome that's B97 and one chromosome that's B73. Then after a number of generations of self-pollination and single seed descent, we start to see these mosaics that are created across the genome. So on one chromosome, individual 1 might look like this, individual 2 might look like this, individual 200 looks like this. And that dissection, that breaking down of the genome and tracking the markers and phenotype is how we're going to identify where the loci are. Now, what's really nice here is you have a number of different populations, and you can actually use the power across populations to better resolve your QTL. Okay, so you can do QTL mapping within one of these, and then you do nested association mapping across these. So there's some advantages of being able to screen more than one allele at a locus. Historical recombination gives you better resolution. There's some disadvantages. This is a lot of work. As you increase the number of individuals, that's more to phenotype, that's more to genotype, That's more to create, more to maintain. And so this is a lot of work, and I don't recommend that people do too many of these for fun. Selective genotyping is another technique people sometimes use. There's some advantages to this and also some disadvantages to this. So in this case, you would take your population and just genotype the shortest 25 individuals and the tallest 25 individuals and use those in genetic mapping. Some of the problems are that there's no genetic map and no normal distribution, and these might be the shortest 25 individuals, but they may have random flowering times. And so if you want to map more than one trait, you can't really do that in this case because you only genotyped those that were the shortest and tallest for the trait that you were most interested in. Bulk segregant analysis is sort of similar. And the reason I really wanted to highlight these two types of QTL mapping, if you want to call it that, it's a marker discovery tool, is because less resources are needed for both of these approaches. So if you're working in an institute that may not have a lot of resources, these are something to consider, but also make sure you consider the drawbacks and don't get too enamored with doing the study if it's not going to give you results that are actually going to be useful or meaningful. So bulk segregant analysis, in this case, you actually bulk DNA from the shortest 25, for instance, plants, or the tallest 25 plants. And so this means you're genotyping only 2 samples, really. I would recommend doing replicates, and I would recommend thinking about this a little more deeply first, but it dramatically decreases the amount of genotyping you have to do. And so this article here, Liu et al. In Genetics in 2010, is an example of how bulk segregant analysis using next-gen sequencing can be used to identify a genetic mutant. Okay, so again, this is going to work well for a major effect gene, a disease resistance gene, for instance. You can have your pool of resistant individuals and your pool of susceptible individuals, right? It's not going to work really well for a trait like yield that's complex and quantitative. Selective phenotyping, I think I'll skip over that. I will just say, just to make you think a little bit more broadly about QTL mapping, one of the things, I would say a mistake that some of us, including myself, made a lot more often was to do our genetic mapping in inbred lines. And ultimately, inbred lines are not a good representation of the variation that a grower would see in a hybrid. So in this case, what we've started to do was do our QTL mapping and our GWAS in test cross hybrids. So you make a segregating population of inbreds, you genotype those, but then you only phenotype those populations on a relevant tester. And this study here, which was a Melchinger study back in 1998, showed very well that there was little overlap between QTL, between the inbred lines and the test cross hybrids. And I would argue the test cross hybrids are the ones we care about. Okay, so just some closing remarks. There's going to be lots of QTL for every trait. And in any one biparental population, only some of those are going to segregate. So that's one of the reasons that markers do not work from population to population in linkage mapping. The largest QTL will be the easiest one to find. It's also probably the one you really care about. Now you can detect a lot of smaller QTL if you increase sample size, but you may not have the ability to do anything with a marker, with a molecular tool to actually select it. Sample size is a limiting factor. It's not genetic markers. It's a common misconception. Increasing our sample size is the best way for us to detect smaller QTL and more accurately detect QTL. Better phenotyping or more phenotyping of the same individuals will also help. But adding more markers, I mean, beyond 5,000 markers, it's not really all that useful. Most traits show 1 to 10 QTL, and each of those QTL can contain 200 to 1,000 genes. So we're not identifying genes in any way, shape, or form in this. So you're not going to find the gene. That's a big misconception that a lot of students have coming into my class. You're never going to be able to detect all of the heritability, and there's lots of reasons for that. That's another 4 classes to talk about why you won't detect it. But you need to have some heritable variation to detect anything using QTL mapping. Transgressive segregation works, and epistasis, although it's not usually detected, is usually the biggest problem that we have. Okay, I'm just going to say a couple words on cloning QTL. So why do we want to do this? If you wanted to have a perfect marker for a gene to use in marker-assisted selection, it's a really bad idea because we're talking about hundreds of thousands or millions of US dollars. However, if your goal is to transform something into another organism, to make a GMO or do gene editing, we first need to identify the causal polymorphism or the gene. Similarly, if you wanted to knock it out or turn it off or overexpress it, we need to identify the gene. We can't just identify a 100 or 200 gene chromosomal locus segment. Now, there's some other advantages. We might want to identify the genetic pathway. For instance, what's the genetic pathway for stem sugar accumulation? We could probably do a lot of biochemistry and start to figure it out, but maybe identifying a QTL for stem sugar might make more sense. To compare across species, so what do these genes do across different species? And then to look at natural variation. So are there other alleles? Once you identify the gene, you can track the alleles, right? So a single QTL analysis is, I'm going to be blunt. I would say it's, well, if I wasn't being blunt, I'd say it's inconclusive. But to be blunt, I'd say it's misleading. What we actually need oftentimes is many QTL studies across environments, across populations, across different researchers to identify where major effect quantitative locus segments are. And so this is just an example from Randy Weiser, Peter Ballantyne, and Rebecca Nelson, combining a lot of different maze studies, I think 50 different disease resistance studies, to look at if there's regions of the genome that correspond to broad spectrum disease resistance. The same authors, or at least 2 of the same authors, did this in rice as well, and then they started identifying candidate genes that might underlie those disease resistance gene clusters. So I'm going to end that first segment on QTL mapping there. The next thing I'll talk about will be GWAS and genomic selection, marker-assisted selection, and then genomic and phenomic selection. And those will be a little bit quicker because I think I've made my major points about our trustworthiness into QTL mapping.